Thank you for being here. On behalf of my co-authors, I want to tell you about the pipeline we designed for the patient-specific prediction of electrode bending in stereotactic neurosurgery. I will start by briefly describing the clinical context and stating the problem before showing our methodology and results. Stereotactic neurosurgery is a minimally invasive form of surgical intervention that relies on optimal surgical planning to target specific regions of the brain. The current pipeline of life-threatening, drug-resistant epilepsy patients consists of identifying potential seizure onset zones and identifying safe trajectories whereby electrodes can be implanted on the patient to sense brain activity. Single regions in non-eloquent areas congruent with symptoms are typically candidate for resection. Implantation accuracy is therefore necessary to ensure safety and efficacy. The problem is that electrodes bend from planned trajectories. In this figure, you can see two examples where bending is observed proximal to the bolt and closer to the tip. Let's have a look at the latter, where a rigid trajectory in the direction of the bolt aims to reach a target point. Often, a rigid stylet short from target point is also implanted. You can also see the last anatomical boundary crossed by this electrode. However, when identifying electrode trajectories from post-operative CT scans, implanted trajectories deviate from planning. And we can observe that the electrode bending direction changes as the electrode bends due to small local displacements along the trajectory that become particularly large when electrode bending occurs. Therefore, being able to account for electrode bending during surgical planning could further optimize accuracy and safety. Our main motivation is to predict electrode bending in patient-specific SEEG electrode implantations. And the aim of this work is to assess two machine learning approaches and two types of labels. Our contributions include an image-based approach that overcomes limitations of feature-based models, an investigation of regressing local displacement versus bending direction, and a fully automated pipeline that is publicly available to the scientific community. Let me briefly describe our methodology. In relation to our pipeline, we first rigidly co-register a post-operative CT image, a T1 weighted MRI image, and a brain parcellation in T1 space before identifying and interpolating electrode trajectories. We use an affine transformation to register T1 images and electrode trajectories onto M&I space. Then we load all the available cases in M&I space and generate data necessary for training in the form of pictures and image windows along electrode trajectories that are used as inputs, as well as two vectors that are used as labels. Once pre-processing is done, we use inputs and labels to train a fixtures-based and an image-based model. Last, we use trained models for inference, whereby an initial trajectory from planning is used to iteratively infer the remaining of the trajectory up to a given electrode depth. And we have also built visualization components to understand electrode bending and predictions in the context of anatomy. Patient-specific cases were acquired at the National Hospital for Neurology and Neurosurgery in London, UK, whereby AdTech SEEG electrodes were implanted. Our dataset consists of 852 electrodes related to a total of 86 cases. And these electrodes were stratified into 6 groups based on brain structures of the entry and target points. We illustrate an implanted electrode to define levels of regression. First, we define electrode direction, VI, as the unit vector projected from previous two contiguous interpolated points onto XP. Next, we define electrode bending, EB, as a unit vector from Xi to the next interpolated point. If the electrode has no deviation, the next interpolated point will be co-located at XP, and therefore VI and EB will be aligned. We can also represent electrode bending as the local displacement LU from XP to the next interpolated point, being 0 if no bending was observed. And note that LU is much smaller than the unit vector EB. We compared two machine learning algorithms. First, a features-based approach where we compute a total of 96 features related to electrode implantation, bending, anatomy, and collision at each interpolated point. And we use these features as inputs to a neural network model composed of 3 hidden blocks and a fully connected layer. Our second proposed method is an image-based approach where inputs are comprised of the electrode direction VI and an image window at interpolated points. And we study windows constructed from T1, GIF pixelation, and a simpler image containing masks referred here as CWD, corresponding to cortex, white matter, and deep gray matter. The image window passes through a convolutional and a max pooling layer. These input layers are stacked into 2 sequential blocks before a fully connected layer is used to infer the outputs. And we use the Adam optimizer to minimize the mean square error loss function. During inference, we regress either local displacement or electrode bending to compute the next interpolated point up to a specified depth. And we also use the dropout layers to compute epistemic uncertainty of our predictions using MC dropout over a total of 204 passes. In this work, we studied the predicting performance of local displacement and electrode bending in handcrafted features and image-based models. And we evaluate the benefit of quantifying uncertainty and study 3 distinct types of inputs for the image-based approach across 6 anatomical regions. We perform tenfold cross-validation and report the MSE of label predictions and MSE of the inferred trajectories. Let me show you some of our results. We summarize tenfold cross-validation in this table. Here, we compare the predictive capabilities of local displacement and electrode bending labels across anatomical regions for a feature-based and an image-based model. The latter having as inputs either a T1, a GIF, or a mask of main brain structures. First, notice that the MSE is lower for LU than EB labels, since LU is much smaller than the unit vector EB. For a features-based model, we observe that bending prediction is the best in electrodes implanted in the inferior frontal orbital gyros, and that there is no benefit of using MC dropout. In contrast, an image-based approach that takes as inputs a window from a T1 image outperforms features-based models across all anatomical regions, with improvements of nearly a factor of 10 when regressing local displacement. We observe improvements using MC dropout in LU and EB labels. However, when we infer trajectories, only 24.2% of the predictions had an MSE below 1 millimeter when using electrode bending, compared to 82.3% when using local displacement. And although performance degraded when using a GIF pixelation as input, we observe improvements in half of the regions when regressing LU when taking a mask of the main structures as inputs. When inferring trajectories, this model performed the best, with 86.9% of the electrons having an MSE below 1 millimeter. For the best performing model, we infer trajectories and compute MSE. On average, across regions, the anterior-posterior central gyros was the group with the lowest MSE, whereas electrodes predicted for the parietal occipital lobes had the highest errors, followed by the middle frontal gyros group. Here, we set the threshold of 1 millimeter that was used to compare model performance. When we look at the performance across regions and folds, we noted that the first and second folds were not as good as the rest of the folds. If we consider only the remaining 8 folds, the means across regions are considerably reduced. And to illustrate trajectory inference of specific regions, we plot the rigid trajectory in white and the ground truth in yellow along the predictions of each fold, with colors corresponding to those used in the plot. If predicted trajectories have an MSE above 1 millimeter and bend in the direction of the ground truth, we color them in blue, indicating that predictions were overestimated. Conversely, if predictions have an MSE above 1 millimeter, but bend in the opposite direction of the ground truth, we color them in red, indicating a wrong prediction. Here we show some good and not so good examples of predicted trajectories using tenfold cross-validation across all regions. To sum up, we presented a fixtures-based and an image-based model for predicting electrobending, and the best model we observed is an image-based model that regresses local displacements and uses an image containing only the masks of the main brain structures as inputs. There are different areas where our work could be extended by assessing the generalizability of our models to other electrode manufacturers and surgical centers, and by applying our proposed models to other stereotactic interventions, and by incorporating electrobending prediction into surgical planning. On behalf of our School of Biomedical Engineering and Imaging Sciences at King's College London, and our clinical partners at the National Hospital for Neurology and Neurosurgery, we would like to thank the funding bodies for supporting our work, the reviewers for their valuable comments, and IPCAI for the opportunity to present our research. Please get in touch if you have any further questions, and thank you so much for listening.